In this video, we will talk about one of the main visualizations or tools in phylogeography, which is bifurcating trees. So remember, phylogeography studies are interested in evolutionary relationships, and trees are one specific way to estimate and visualize those relationships. So in general, bifurcating trees, or I'll often just call them trees, reflect a series of branching processes where one lineage splits into two or several descendant lineages. So this is the same tree graphic I've shown before. And the main thing to note is that the positioning of taxa on a tree is based on their genetic similarity. So what that means is that if we're looking at these individuals from Fiji, for instance, they're situated next to each other on the tree. That means they're more genetically similar to each other than, say, one of these individuals from Fiji and this one from the Red Sea. In addition, these individuals from Vanuatu and Fiji are more closely related to each other because they're more close to each other on the tree. That means they're more genetically similar to each other than either of those are to one of these individuals from the Great Barrier Reef, for instance. So overall, what we're looking at here in these trees are branching processes. So this is one example of one lineage that split into two, and then from there you end up with these endpoints, and what you're looking at is essentially clustering at these endpoints. So if these lines are clustered together, that means they're more genetically similar, more closely related to each other than other individuals or populations that are farther apart, farther away distance-wise on that tree. And there are several different ways to make trees. We're going to focus on trees in this class constructed from genetic characters. So this is one example of a tree built from this paper that looks at different mammal species. And specifically, this tree is pulling out and focusing on marine mammals. And the reason why I mentioned that we're looking at genetic characters is that you can build trees based on morphology, but the trees you might build based on morphology doesn't always match the tree you might build from genetic characters. So if we're thinking about marine mammals, for instance, marine mammals have these flippers, they have the flukes. So morphologically speaking, they look relatively similar. But if we build a phylogeny based on their genetic characteristics, their genetic sequences, they're not all that closely related. So we do see some instances where bottlenose dolphins and orcas are closely related because they're close to each other on the tree. But if we look at walruses, for instance, they're way down here, and walruses are actually more closely related to dogs than they are to any other marine mammal. And manatees are also very closely related to elephants, which obviously not a marine mammal. So if we look at this tree, again, we said this tree is based on genetic characters. This down here is our scale bar, essentially. So the longer one of these lines is, the more evolutionary time or evolutionary distance that represents. And so this scale bar is telling us that this particular distance on the tree is representing 0.1 nucleotide substitutions. So again, the longer that line is, the more nucleotide substitutions there are between, say, this old lineage and what it's split into. So again, remember, if it's more close together on the tree, more closely related, genetically speaking, because we're building these trees based on genetic characters. And there are several different methods to build these types of trees. We specifically are going to focus on methods called maximum likelihood, often abbreviated as ML, to reconstruct these phylogenies from molecular data. And so the nitty-gritty statistical details are outside the scope of this class, but in general, the way that this process works is you build several different possible trees. So these trees might differ in their branch points or their lengths of branches, so that's one possible tree. This one might have a shorter distance there, longer distance there. Anyways, I don't know, these look like sad, weird cacti or something. They're supposed to be trees. The idea is that you make a bunch of these different possible trees based on the molecular data that you have. They have different branching patterns, different branch lengths, and then through these statistical techniques, that's what the actual maximum likelihood methods are. Again, outside the scope of this class, I don't expect you to know the details, but these maximum likelihood methods are then used to determine which of these possible trees is the one with the highest total probability of being correct. So say we have these different trees, and maximum likelihood actually decides that this one has the most chance of being correct. Because you're building all of these different trees and then choosing the highest probability one, this maximum likelihood method is relatively computationally intense. This is one example of a maximum likelihood tree constructed for different Drosophila species. And one of the main things to point out in these trees is they contain something called bootstrap values. So if you look at the branch points in this tree on the right, you see these numbers that are listed here. Those numbers are the bootstrap values, and a higher bootstrap value indicates a higher confidence in that branch point in the tree. So remember, we said for maximum likelihood methods, you're basically creating several different possible trees and then choosing the one that has the most likelihood of being correct. The higher this number is, that means even across these different potential trees you were building, a lot of them had this same branch point here. So if a lot of the potential trees had this same branch point, we're pretty confident that this branch point is correct because it showed up in many of the different possible trees. So bootstrap values go from 0 to 100, where 100 mean, yes, 100% we know this branch point is correct. Generally, you don't see numbers that high. Often above like 75-ish is considered a really good value that we're confident in. So if we look at this particular tree, we see some 90s, 97s, we're really confident in those. We also see some lower ones too, so 41, so that we're not super confident in that particular branch point. So that's something to think about as you're looking at these trees. Make sure you're looking at the bootstrap values as well. Even though a branch point is there, if it has a relatively low bootstrap value, we might not be super confident that that's actually an accurate branch point. Okay, and so then what I want us to think about now is how these trees can actually build on things we've talked about previously. So before, we've talked about neutral versus adaptive markers. What I want you to do is refresh your memory. What is the difference between neutral and adaptive markers? That's the first thing. And then secondly, think about, do you think trees constructed from neutral versus adaptive markers will look similar or different, and why? So I'll give you some time. You can pause the video if you need to think about those two questions before we move on in the rest of this video. Okay, so remember, neutral markers are markers that do not affect fitness. Adaptive markers are in genes that do have some direct effect on fitness. And if you construct maximum likelihood trees from either neutral or adaptive markers, you actually get different results. So the reasoning, these are two different trees. I'll go into more detail on these in the next slide, but these are both maximum likelihood trees. One of them is from neutral markers, one of them is from adaptive markers. They kind of look like fireworks, but if you look at the colors, there are different patterns there. But the reason why neutral and adaptive markers give us different results is, again, if we think about neutral genes don't affect fitness. And so because of that, generally, you can have similar sequences between different populations, which means there are no distinct patterns and relationships on a tree. In contrast, when we think about adaptive markers, the ones that do affect fitness, we would expect those particular markers to then show corresponding differences and patterns in different populations exposed to unique environmental challenges and unique habitats, for instance. And so if we explain here what we're looking at with these trees, these are SNP trees, so these were conducted from, in particular, SNP markers were used for these trees. They're from the snail species that I study. And again, these were constructed from, here on the left, we have the neutral markers. So really this was constructed from all possible SNP loci that I identified through RADseq. This was about 1,600 SNPs, I think. Because the vast majority of SNPs are neutral, the all loci tree can also be considered the neutral tree. This one here on the right was constructed from adaptive, what are called outlier loci. So they're outliers because the genetic differences at those particular markers are larger than average between the different populations that we're interested in, for instance. And so when we look at these trees, to orient us, each branch tip is an individual. So this is one particular individual. The colors at each of those branch tips represent whether that individual was from Southern California. So if there's a red square, that means it's from Southern California, and then a blue square is Northern California. And so overall, if we look here, the patterns are different for these different trees. And what I have here at the top, it says only SNP trees constructed from those outlier loci show population differences. So what I mean by that is if you look at this tree on the left, first of all, you may notice my Bootstrap values are terrible. So take this with a grain of salt. But overall, if we look at this tree on the left here, constructed from all loci, most of which are neutral, we don't see any patterns. So basically each one of these branches were seen as a mixture of individuals from both Northern and Southern California. But if we look at the tree on the right, which is from the outlier loci, we can see some patterns here where this branch, this group is all Northern California individuals. Here we have a lot of Southern California individuals. So when we're using only those adaptive loci, we're starting to see differences emerge between the populations. And given what we know about these different populations from what I've talked about previously, what do you think might be some of the functions of these outlier loci? What kind of gene functions might these loci that have large genetic differences between populations, what might some of their functions be? What do you think? So briefly, I could talk about this for a while because I think this is maybe one of the most interesting things that came out of my dissertation. But these adaptive outlier loci have functions related to heat stress tolerance. So remember, in Southern California, it's warmer, Northern California, it's cooler. And specifically, a lot of those outlier loci I found are involved in something called stress granule formation, which are formed to help individuals survive stress, such as heat stress. So I'll leave it at that because this video is already getting a little bit long. But if you want to know more about that, please ask me. I'd be happy to talk more about it with you. Okay, so to wrap this up here, overall, trees are good for looking at relationships among different species. So again, if you wanted to compare these different species of Drosophila, trees would be a good way to do that. However, trees are not always good if we are specifically interested in looking at different populations of the same species. So these individuals are all the same species of fruit fly. If we wanted to compare these different populations, a tree might not be appropriate for that. And that's because in many cases, the genes that we're using to build these trees might not be different enough to actually have patterns on trees that are informative for us. So the next video we'll talk about, if we can't use trees for these types of populations, what is a good alternative approach to take instead.